Hello guys, you're probably still sleeping. It's now 9 a.m. Hope you're enjoying the day of rain and no school. Today we're going to learn a technique used to replicate or make many copies of DNA. Previously, the only way to make copies of DNA of a specific sequence that you want was to find the gene of interest, cut it with a restriction enzyme, put it in bacteria, and then the bacteria would replicate the DNA for you. You're going to see this week that it takes multiple days, and it's actually rather smelly as well. So they've discovered or invented or designed, I guess, an easier way to make copies of DNA. This method is called PCR, or Polymerase Chain reaction. Kaiser, get out of the way. And it's a method for making many copies of DNA. And you actually only need one copy of the sequence, and it will replicate the sequence many times. You can get millions and millions of copies of this one sequence of DNA. So you no longer need to use E. coli or bacteria to replicate the DNA. We now have this method called PCR. It's really a machine, which you're about to see in a second, that will replicate the DNA for you. In this lesson, we're going to go through the steps and the requirements for replicating a strand of DNA. So, PCR occurs basically in a test tube and a machine that cycles through temperatures. In the test tube, the test tubes are really small. They're actually even smaller than the ones I showed you the other day. About, I don't know, half the size of that one, maybe even smaller. Is you need the template strand, or the strand that or the DNA that you're trying to replicate. Okay. You need the DNA polymerase enzyme. Remember, DNA polymerase adds corresponding DNA nucleotides to the template strand. So, once again, you need the strand of DNA you want to replicate, which you can cut out of an organism, extract it from an organism, and you have it. Then you need the DNA polymerase enzyme, which is going to replicate the DNA by adding the corresponding nucleotide to the template strand. Then we need nucleotides to add, so the A's, the G's, the C's, and the T's. And then the most important thing, and the thing we're going to talk about mostly in this lesson, is you need a primer, okay, to start replication. If you remember way back in, I guess that was December, we discussed replication. It might have even been November, to be honest. I think it was before Thanksgiving. DNA polymerase, our good friend DNA polymerase, cannot add to nothing. It needs something to add to. So we need to add or design primers, and primers are small segments of DNA that flank either end of the DNA that you're going to replicate. So just to recap, you need a template strand. This is the strand you're going to replicate. You need the DNA polymerase enzyme, which is actually going to do the replicating. You need the nucleotides so that they can be added by the DNA polymerase enzyme. And then you need the primer, which is going to mark the DNA you want to replicate on either end, so that DNA polymerase can add to something. Okay, so once again, as we said, the primers are critical. Okay, you could have this really long stretch of DNA, and you only want to replicate some of it. The primers are going to mark either end of the sequence you want to replicate. So if you look at this picture here, this is the DNA we want to replicate. We design a primer that's complementary to the sequence on one strand, and another primer that's complementary to the sequence on the opposite strand. And then DNA polymerase will come in and fill in the bases here. Notice once again that it's going in a 5' to 3' direction, adding to the 3' end, but going in a 5' to 3' direction. So primers, basically, they identify which sequence they bind to either end of the sequence on either strand that you're going to replicate, so the DNA polymerase can add to the primers. So let's say this was the strand that we're going to replicate. This just means that there's lots of DNA in here. I didn't feel like drawing lots of letters. You would want to design a sequence that's complementary here. Let me get the pen out. That's complementary to, like, say, this sequence here. So the primer would therefore be T, A, T, A, T, and then you want to design a primer on the other side of the sequence that you want to replicate here. And obviously you guys can figure out what the primer would be here. By adding the primer, that signals that DNA, this is where DNA polymerase should bind and start adding base pairs in this direction and in this direction. Because if you remember, DNA polymerase can only go in a 5' to 3' direction. Okay. So now that we have those things in the tube, okay, in the tube we have the DNA you need to replicate. Okay, you have the primers that tell where DNA polymerase should start doing the replication. You have DNA polymerase, and you have the nucleotides all in a test tube. Okay, you're now going to put it in a machine, which we actually have one at school. We're not going to use it because we don't have time. But you're going to put it in a machine that cycles through temperatures. Okay, and it cycles through 3 different temperatures that allow the DNA to replicate. Okay, we're going to talk about those 3 different temperatures and what they do to the DNA that allow it to replicate. Okay, so first, the machine heats up to 90 degrees, and that allows the DNA strands to separate. That it sort of denatures the DNA or unwinds it. Okay, why do we now? Who do that normally in replication is DNA helicase, but in this case we don't have DNA helicase. We can't be throwing all these enzymes into the tube. So we're going to denature or unwind the DNA using heat. Then we're going to cool it when the machine's going to change temperatures and cool the DNA to about 55 degrees Celsius. Okay, that allows the primers to stick or hybridize, the word is hybridize, to either end of the DNA that it's going to replicate. If it was too hot, they wouldn't be able to stick. So we're going to cool down the DNA in the tube to 55 degrees Celsius so that the primers can stick. And then last but not least, once the primers are stuck, we're going to heat up the DNA to about 70 degrees Celsius. And that's going to allow DNA polymerase to add the corresponding bases after the primer. And this will continuously cycle through these 3 temperatures again and again and again and again until you have about 1 million copies of this one DNA sequence that's marked with primers. All right, so this is a picture showing the same thing. So let's say you have a drop of blood. Okay, there's DNA in this blood, and you want to maybe amplify a certain sequence that's in that blood. You're going to design primers on either side of the DNA sequence you want to replicate. Okay. Then you raise the temperature to 90 degrees Celsius. You put it in the tube. Let's actually take a step back. Put it in the tube with the DNA polymerase, the primers, the nucleotides, and then you put it into the machine. Okay, the machine is first going to heat it to 90 degrees, where it's going to separate the 2 strands. Okay, then it's going to cool it back down to about 55 degrees. That's going to allow the primers to anneal to either side of the strand that it's replicating. And then last but not least, you're going to heat it back up to about 70 degrees, and that allows DNA polymerase to add the corresponding bases. And as you can see, you get about 1 million copies of one sequence in about an hour, as opposed to replicating in plasmids, which takes multiple days, or bacteria, which takes multiple days to do. Okay, now, you might have thought about this, maybe not, is that most enzymes denature at high temperatures. So in order for this process, the person who was designing this, who they're now a millionaire, I'm sure, they needed to. If you have the problem that every single time you heat it to 90 degrees Celsius, which is really hot, the enzyme's going to denature. Then every single cycle you would have to add a new enzyme, and that would make the process really, really slow. You know, cycle through the 3 temperatures, take the tube out, and add the enzyme again because the enzyme's now ruined; it's denatured. It doesn't really make the process any faster. The purpose of this machine was to make the process faster. So they needed to find an enzyme or a DNA polymerase enzyme that could withstand warm temperatures, and they found one in a bacteria that lives in hot springs, so really, really hot temperatures. Its DNA polymerase is called Taq polymerase, and it can withstand temperatures of between 90 degrees and higher. So you don't have to worry about the enzyme denaturing. Now, how is it different from our polymerase? Is it probably has a different amino acids which make it fold differently and make it be able to withstand temperatures that our DNA polymerase cannot. This allowed the process. I mean, this was basically the whole like the key discovery in being able to do PCR is finding a DNA polymerase that could withstand warm temperatures. Okay. So this process is long, and in terms of sort of like the process itself is not long, but like what is the benefit of having this process? Like what do people use it for? And I mean, the first sort of major thing that it was used for is that you can get a large amount of a DNA fragment. And so if you want, for example, if you have the insulin gene and you want to put the insulin gene into plasmids, you need many copies of it to put it into plasmids to make the bacteria accept it. The more copies you have it, the more likely it is to go into the plasmid. So if you design primers that mark either end of the insulin gene in a human, you will replicate and only get the gene for insulin, and then you can put it in plasmids. The second reason why it's used is that if you find DNA at a crime scene, a lot of times it's in a small quantity, not enough to do testing on. So you can stick the DNA that you find into a PCR machine and replicate it to get many more copies of the DNA so you can do testing on it. A lot of times lawyers, they'll like raise the fact that the DNA found at the crime scene was a small amount, and they'll still like sort of pick at the fact that it was replicated with PCR, but PCR is actually generally pretty accurate. And the last, and this is sort of the more recent uses of PCR, is that you can determine if a particular sequence that you're looking for is in a DNA sample. So if you remember, in order for this to work, you have to design the primers correctly. And if the primers are not correct, they don't bind anywhere in the DNA, then the replication won't work. So if you're looking for a particular sequence in a DNA sample, okay, you can design a primer that has that sequence, okay? And if that sequence is in the DNA, well, the primer actually would have to be complementary to the sequence. And if the DNA has that sequence, the primer will bind, and the PCR will work. And we're going to talk about next week how you can tell if the PCR works or not. But if the sequence or your DNA sample does not have that sequence, the primer won't bind, and then PCR will not work. So you can just tell whether or not the PCR works by designing a primer to the DNA sample if the DNA sequence that you want to see is in your sample. If it works, the primer will bind, and then you'll get DNA, or the PCR will work. And if the primer sequence is not there, then the PCR will not work. And then you know that that sequence is not in the DNA. Tomorrow in class, so tomorrow is a full day of school, it's not a half day. Tomorrow we're going to talk a little bit more about this. And talk about what we're going to do in the lab, which I think now is going to be on Thursday. I'm not totally sure of the schedule yet. But I will see you guys tomorrow if it stops raining. I don't know. I mean, it could be dangerous out there. There could be rain. All right. Have a good day, guys.